Hi everyone. Why do we age? It's one of the fundamental questions in biology. We often think of aging as a simple process of wear and tear, but the full story is much more complex. Today, we're going to examine the ultimate evolutionary causes of aging. We will move beyond the simple mechanics of how bodies break down to explore the evolutionary logic of why this process occurs at all. To properly investigate this question, we must first make a critical distinction. It's essential to separate the immediate biological mechanisms of aging from the long-term evolutionary drivers. This is the difference between asking how a machine fails versus asking why it was designed in a way that makes eventual failure inevitable. So, this brings us to the concepts of proximate and ultimate causes. The proximate causes are the how. This is the focus of most aging research. Mechanisms like cellular senescence, DNA damage, and telomere shortening. The ultimate causes, which are our focus here, address the why. They are the evolutionary pressures that have shaped the aging process over vast timescales. Now, in an evolutionary context, survival is not the ultimate goal. The goal is to maximize fitness. Fitness is a composite measure, and we can break it down into four key components. First, an organism must survive to the age of reproduction. Second is the number of descendants it produces. Third is the generation time, or how quickly it can reproduce. And fourth, a component that is often overlooked but is critical, is the quality of those descendants, meaning their own ability to survive and reproduce. A multitude of theories have been proposed to explain aging. To make sense of them, we can categorize them into three broad, mutually exclusive frameworks. This organizational structure provides a clear way to understand their relationship to evolutionary forces and, importantly, allows us to test their predictions against empirical evidence. The logic for classifying these theories is quite straightforward. We start with the first question. Does the theory invoke evolutionary forces at all? If the answer is no, it falls into our first category. If the answer is yes, we then ask a second question. Does the theory posit that aging is a consequence of a failure of these evolutionary forces? The answers to these two questions allow us to neatly sort any theory of aging. Our first category contains the mechanistic theories. You can think of these as the wear and tear hypotheses. They propose that aging is simply an unavoidable byproduct of physics and chemistry, that organisms, like machines, inevitably break down over time due to processes like oxidative damage. In this view, evolution is either irrelevant or powerless to prevent this decline. Category 2 also frames aging as a failure of evolution, but for a different reason. This is the idea of the weakening force of selection. Since extrinsic factors like predation and accidents remove individuals from a population over time, there are fewer older organisms. Consequently, natural selection is less effective at purging mutations whose harmful effects only manifest late in life. These mutations are thought to accumulate in the genome, leading to senescence. This brings us to the third category, optimization theories. This framework represents a significant shift in perspective. It proposes that aging is not a failure or an accident, but rather an evolved strategy. The core idea is that organisms must allocate finite resources between somatic maintenance, that is, survival, and reproduction. Evolution, this theory posits, has optimized this trade-off to maximize overall fitness, not merely to extend lifespan indefinitely. Okay, so we have our three distinct frameworks. Now we must evaluate them based on their ability to explain real-world observations. We will test them against 5 key pieces of empirical evidence from biology. A valid theory must be consistent with these findings. This table provides a summary of the results. As we'll discuss, the mechanistic and weakening force theories are inconsistent with several key observations. The optimization theories, however, appear consistent with all of the empirical data we will examine. Let's now walk through the evidence for each test. Our first test is caloric restriction. It is a well-documented finding that reducing caloric intake extends lifespan in a wide range of species, from yeast to primates. This observation poses a significant problem for the first two theories. Mechanistic theories would predict a corresponding reduction in metabolic rate, which is not consistently observed. The weakening force theory suggests that lifespan should be difficult to modify so substantially. Optimization theory, however, provides a clear explanation. During periods of famine, it is evolutionarily advantageous to divert resources from reproduction towards somatic maintenance, effectively waiting for more favorable conditions. Next, consider the existence of species with slow or even negligible senescence, such as certain turtles, bats, and the freshwater hydra. Mechanistic theories struggle to explain, for example, why bats, which have a high metabolic rate, are so long-lived. The weakening force of selection theory is also inconsistent with certain findings, such as cases where higher juvenile predation selects for longer-lived adults. Optimization theory accommodates these observations by predicting that lifespan is optimized based on the specific mortality risks an organism faces throughout its life history. A demographic finding that is perhaps surprising is the existence of mortality rate plateaus. In many species, including humans, the exponential increase in the risk of death with age slows down and levels off at very advanced ages. This directly contradicts a core mathematical prediction of the mutation accumulation theory, which posits that mortality rates should increase exponentially without limit. Mechanistic theories also fail to predict this phenomenon. Optimization models, however, are capable of generating such plateaus. For a particularly stark example, we can look to eusocial insects. A queen ant or bee, despite sharing the same genes as the workers, can live more than 30 times longer. This occurs while she maintains an enormous reproductive output. This observation is fundamentally inconsistent with simple mechanistic theories that link aging to metabolic expenditure or wear and tear. From an evolutionary standpoint, the queen is highly protected from extrinsic mortality, creating intense selective pressure to invest resources in longevity to maximize the colony's reproductive success. The evidence we've reviewed so far points strongly toward optimization theory. However, this framework makes a unique and critical prediction that sets it apart from all others. Testing this prediction provides the most stringent evaluation of the theory. And here is that key prediction. If species in their natural environment are at or near a fitness optimum, then any single mutation that extends lifespan must come at a cost to one of the other components of fitness. The other theories view aging as purely a defect, so fixing it should only have positive consequences. Optimization theory, in contrast, predicts a necessary trade-off. For some time, this prediction appeared to be falsified. Seminal experiments in the nematode worm C. elegans showed that mutations in a single gene, DAF2, could double lifespan without any apparent cost to the number or timing of offspring. This phenomenon, termed longevity for free, presented a significant challenge to the optimization framework. The resolution to this paradox is found in the fourth component of fitness, offspring quality. Consider the Pacific salmon. They are semiparous, meaning they die after a single reproductive event. This is an adaptive strategy. Their native streams are nutrient poor, and their decaying carcasses provide a critical source of nutrients for their own offspring, thereby directly investing in their quality and chances of survival. A similar mechanism has since been discovered in C. elegans. After reproduction ceases, wild-type worms repurpose their intestinal tissue into yolk, which is then consumed by their larval progeny. This is a form of parental investment that enhances offspring quality. The long-lived DAF2 mutants are deficient in this process. When placed in direct competition, the fitness of these mutants is only about 35% that of the wild-type worms. The fitness cost was not absent, it was simply hidden in the fourth component of fitness. With this paradox resolved, we can now synthesize the findings and consider their implications for future research into the biology of aging. When all the evidence is considered, optimization theory emerges as the only framework consistent with the full range of empirical data. It reframes aging not as a failure or an accident, but as a trait that has been shaped and optimized by natural selection as part of a complex series of life history trade-offs. This conclusion has significant practical implications. It suggests that lifespan is a highly regulated and malleable trait, not a fixed, intractable process. This supports the idea that interventions targeting the biological pathways that manage these trade-offs, such as nutrient-sensing pathways, are a promising avenue for research. It also lends strong theoretical support to the geroscience hypothesis, that by targeting the aging process itself, we may be able to delay the onset of multiple age-related diseases. This leads us to the next frontier. A critical direction for future research is to design experiments that can accurately measure the fitness costs and trade-offs of longevity-extending interventions under ecologically relevant conditions. Moving beyond the controlled environment of the laboratory to understand these dynamics in the wild is the next major challenge for the evolutionary biology of aging.